Hi, welcome to Neuroverse
A podcast hosted by Carolina.
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on Enzyme-Directed Evolution.
Woo!
Yes.
I always feel like there should be a little applause at
Yeah.
that point.
To start off with, I will first just talk a little bit about enzymes, which I think are really cool. Given my degree was in biochemistry, I studied them a lot. So enzymes are biological catalysts. They are proteins which are made up of amino acids, and they help speed up reactions by lowering the energy it takes for a reaction to occur. Enzymes are everywhere in the biological world. An example that is close to us is, for example, when we digest food, our enzymes break down the food. And if we look at other species, this also happens, for example, with mushrooms, except they digest food externally. So they release enzymes to break down organic matter and then absorb the nutrients. But enzymes also do several other things. It's not just about breaking down food, they are also present with DNA transcription and translation, as well as viral encoding. Yeah.
Basically, all biological processes pretty much require enzymes.
Yes, exactly. Yeah. The structure of the enzyme is very important for their function. This is because they have an active site, which is where the reactions happen. And they either have amino acids which are hydrophobic or hydrophilic, and this allows for other molecules to bind and react and assemble or break down. And just to emphasize the importance of structure in biology, I want to do a little side take and talk a little bit about chaperones.
Ooh.
So chaperones are these proteins in our body which help assemble other proteins.
Mm-hmm.
So they'll take a protein that is like uncoiled, that is like in its primary structure,
Mm-hmm.
and help fold it up. And it's so cool. It's like this little, I don't know, encasing where the protein filters its way in, and then because of the hydrophilic or hydrophobic amino acids inside, it then slowly bends the protein into shape. And then once it's folded, it releases it. And
so do you know if the same type of chaperone always makes the same type of proteins into the same shape, or can it make different shapes, basically?
yeah, so it depends. Some chaperones are really like protein specific, so they'll only fold one specific type of protein. Other chaperones are not that specific. They'll just fold proteins in general. And it's not like they think, okay, I need to fold this protein into this shape. It's more like due to the hydrophobic proteins of the chaperone and due to the hydrophilic proteins of the protein, they then like the protein then bends into the shape it's meant to be.
That's so interesting. So the resulting shape is actually a product of the relationship between the two compositions of the different proteins, the chaperone and the enzyme.
Yeah, exactly. It's so neat, and it's kind of simple, but it leads to so many like intricate and like thousands of different types of proteins and shapes.
Yeah, that's what I find most interesting. I don't know nearly as much about enzymes as you do, but it's the fact that enzymes are made of thousands of amino acids, but there are only 20 types of amino acids.
Yeah.
And with that combinatorial type mechanism, you can just create so many different types of enzymes that catalyze all these different types of reactions.
Yes, it's incredible.
It's responsible for the diversity that we have in the biological world.
Exactly. Yeah, diversity is so important in the biological world.
Yeah.
Okay. So, staying within the structure, there's a couple of ways to figure out what the enzyme structure is. In biology, we can use techniques such as NMR and crystallography, which allow us to visualize the protein shape. However, the issue with this is that sometimes you'll get an average of the shape because it takes like snapshots. So it's not the most accurate.
Because the shape changes.
because depending on what angle you look at the protein, you may see something slightly different.
Right, because it's so complex.
Yeah, and like it's 3D, so like it expands in several different ways.
Yeah.
And then another thing you can do is have a computational approach. So using, all that we know about the structure of amino acids, the bonds, the angle of the bonds it makes, its hydrophobic properties, we can model what we think the structure should be. And these techniques, they used to have a rate of around an accuracy rate of 40%, which is very, very low.
Yeah.
So if we want to like pick on these enzymes and develop them further, it would be like, it would just be guesswork.
Yeah.
And so then AlphaFold came into the picture.
I was thinking about that because I only recently read about this. And didn't they increase the success rate to like 80% for the first time?
Exactly.
Yeah.
So in 2016, they managed to increase the accuracy rate to 60%, and then 2020 to over 80%, which is double in like 4 years.
What I found really interesting about this, which is a bit of a is a bit of a tangent, but the reason they were able to do this is because they were trying to win a competition.
Oh yeah.
Because there was like a competition for how accurate you can estimate protein structure, and they won, and that's how they got to. So that's just so a little amazing tangent of how competitions drive science sometimes.
Yes. So this is great because now that we have a much more accurate picture of enzymes, we can then properly like play around with them. Enzyme mutation and evolution and us tinkering with enzymes has always been happening, but now is like the best time because now we have the most accurate picture of them.
Yeah.
And so into this comes Frances Arnold, which won a Nobel Prize
in 2018, I believe.
Yes, exactly. And she wanted to build new enzymes, which is now what I'm going to talk about, enzyme-directed evolution. So nature is incredibly abundant, and like we talked about, these 20 amino acids, they lead to such incredible variety, but we still have problems that we want to solve, and we still have reactions that we want to improve. And so why do we limit ourselves to just the biological world? We could also, there's countless of other enzymes in the periodic table. Why not play around with them and see what we can get out of them? And so this is what enzyme-directed evolution is. It's when you take an enzyme and you want to optimize it to do the reaction that you want it to do. So you mutate it, and then from those mutations, see what works best, and then over generations, achieve the optimal enzyme.
I think the coolest thing about this is that it takes advantage of random mutations or mutagenesis, because as far as I understand, like you were discussing earlier, you can computationally design proteins, but often this is limited in that it just doesn't serve the purpose that you want it to serve, because we're still not able to predict the function of something directly from the structure.
Even though there's a really like high correlation, in practice it's different.
And so it seems like the best way to do so is enzyme-directed evolution, which is basically letting nature do its thing, but sort of guiding it.
Exactly. Like we take nature and like we amplify it, we ramp it up.
And the interesting thing is we don't really know what's happening. We just, because it's based on like the functional output. So once it gets to a stage where this enzyme serves the function you're trying to make it serve, then we're happy. But like whatever's happening between, I guess we don't know the fine details.
Yeah, exactly. Sometimes scientists have, because you change one single amino acid, you change one single functional group, and it can lead to several different outcomes. And so sometimes scientists would just do one mutation at a time, but this takes billions of years if you want to, if you wanted to go through every single mutation, if you wanted to try every single option. But nature doesn't do that. Nature just adapts on the spot a good enzyme, a good a good example is
all enzymes are good. No,
yes,
maybe not all enzymes are good though.
I don't know. Is we've thrown like herbicides on fields,
Yeah.
and this environmental pressure caused nature to come up with an enzyme atrazine degradation, which dechlorinated atrazine. So, and this is evolution in real time, which is incredible. So nature doesn't do one mutation at a time. It doesn't wait billions of years. It just comes up with a solution. So why not take advantage of this? Why not use the enzymes already out there and just tinker with them and quote unquote evolve them to do what we want them to do?
Yeah, but I think an interesting thing is the fact that, well, it's obvious now that not all enzymes that we need for our human purposes have been created by nature's process of evolution. And so, for example, I think enzyme-directed evolution nowadays is used mainly to come up with sustainable solutions of how to break down compounds like plastics. And these sorts of problems haven't yet been overcome by nature. And this is why we need enzyme-directed evolution.
Especially if we want a faster approach. Over time, nature may come up with a solution, but it could take years. Evolution can take years. And we found a way to like speed up the process. So essentially, enzyme-directed evolution is when you have an enzyme that is really good at doing a specific job, but in that enzyme you find an area called a promiscuous region where you are then able to direct it and optimize it to do a different task. So it's a small area that does have some capability of doing the task that you want it to do. And in the beginning, it's going to be really bad at doing that job. But over time, you can optimize it.
Yeah, it's all about optimization.
Yeah, exactly. It's a little bit like us humans. So, for example, we're doing a degree in neuroscience. We want to become neuroscientists and so on.
Yes, we do.
But in a different environmental circumstance, perhaps we would have been artists or engineers. Or something else. And so that's a little bit what this is. It's the same enzyme, but in a different context and with a different pressure, you can get it to do different things.
Right.
And Frances Arnold showed this so incredibly well with her research, and where she works with cytochrome C a lot.
Do you want to just explain what that is?
Yeah, so cytochrome C is a mitochondrial protein, and she was able to, quote unquote, convince it to create a reactive intermediate to carbon to carbene, which is something that doesn't happen naturally. And then she was also able to convince a heme protein to involve. And then Kai, who works with Frances Arnold, also showed that he could make cyclopropane. All of these things don't happen in the natural world. And they also found that they could make carbon-silicon bonds, which is insane. Again, so carbon is the second most abundant element, and silicon is the second most abundant element. And
I didn't even know that.
Yeah.
It's such an underrated element.
It is.
What?
But carbon and silicon, we've never seen them bond before, but they've managed to do it. So cytochrome C heme proteins were isolated from a bacteria found in Iceland. In the right circumstance, these iron-containing proteins were able to evolve to bond to form carbon-silicon bonds. This is an example of how you can incorporate non-organic materials and form them and cause them to form like carbon bonds, which widens completely our repertoire of
kind of like our chemical toolbox.
Exactly.
Yeah, that's incredible.
Yeah. This is what I find most incredible about enzyme-directed evolution. It's amazing how we can optimize enzymes, but what's mind-blowing is how Frances Arnold has been able to create carbon bonds with n- non-organic materials. It just opens the door completely. I'm just in awe of this.
Yeah, so am I. And she seems like such a remarkable scientist.
Yeah.
My gosh. But I also read about, so Frances Arnold won the Nobel Prize together with George Smith and Gregory Winter. And she was in charge of sort of coming up with enzyme-directed evolution as a mechanism. But George Smith and Gregory Winter, they discovered how to get bacteria to make proteins that we want.
Yeah.
And so it's really interesting because George Smith used bacteriophages, which are bacteria infecting viruses, and he made it so that they can make any desired protein. And then the cool part is that they then stick these proteins to their outer coats, and then these can therefore be used by inserting, for example, if you want to make like an antibody in the bacteriophage, and then they will display it on their coat. And this is valuable for creating therapies, basically, because then you can create antibodies to order to target specific proteins for diseases like autoimmune diseases or maybe even some cancers, which is incredible. There's so much potential.
That's amazing. So much potential. And it's, like you said, going back to the bacteria, it's also an amazing way of, if you incubate it, to then just create like thousands of these enzymes. And
it's a very fast process. You can you can amplify it. And I also this whole topic kind of just reminds me of like the human connection with nature. Like I feel science always works best when scientists observe what happens in nature and then actually utilize it, rather than creating a process that's totally human, unnatural, and based on just our logic and knowledge, but instead we can incorporate like natural processes in this way. And it reminded me of one of my all-time favorite scientists, who's actually she's such a polymath, but she's like an engineer, architect, designer, and her name is Neri Oxman. She has a lab at MIT, and she basically created the field of material ecology.
Wow.
So she trained as a doctor and an architect.
Amazing.
But now she's doing a lot of like, her research kind of intersects engineering as well as design, and her lab like creates new biosynthetic materials. And they have one of their projects was creating this frame and then getting a bunch of silkworms and just putting the silkworms on this frame, and it created this huge silk pavilion.
Wow.
And just a lot of projects like this. And every time I listen to her talks, she always ends with the same quote, which is like, we as humans now have a responsibility to mother nature, instead of like, mother nature. Play on words.
I love that.
And I think enzyme-directed evolution definitely fits with this idea of like mothering nature, because we're in a way controlling like natural processes, but also enabling it to have its freedom to do its thing.
Yeah, exactly. And we've like you've mentioned, these human developed tools in especially inorganic materials and things like that, and these two worlds, inorganic and organic, used to be so separate, and we've developed such incredible things in the inorganic world as chemists. And now this enzyme-directed evolution, the fact that we've been able to create these bonds, just completely like opens this gate. And it's like, everything is our oyster. We can access the possibilities. We can access the whole periodic table and not be limited to like 4 or 5 elements, which is what usually happens in the organic world. So I find it fascinating. So we hope you enjoyed this episode. Let us know what you think about these organic and inorganic bonds being formed, and any opinions you might have on enzyme-directed evolution.
We hope you enjoyed learning about enzyme-directed evolution. I know I did. We'd love to hear all of your thoughts, so do contact us through our social media accounts.